Discover how lysosomes create and maintain their acidic environment, and why this is crucial for cellular function. Lysosomes are the cell's recycling centers, maintaining an internal pH of around 4.5, which is about 1,000 times more acidic than the cytoplasm. Let's first understand the basic structure that makes pH maintenance possible. Lysosomes are membrane-bound organelles containing more than 50 different digestive enzymes called hydrolases. The lysosome has a lipid bilayer membrane that separates the acidic interior from the neutral cytoplasm, with hydrolytic enzymes floating inside. Now let's explore how lysosomes actively create their acidic environment. Vacuolar ATPase pumps, or VATPase pumps, are embedded in the lysosomal membrane and actively transport protons into the lysosome. The VATPase pump uses energy from ATP to transport hydrogen ions from the cytoplasm into the lysosome, creating the acidic environment. The pump hydrolyzes ATP to release energy, which powers the active transport of protons against their concentration gradient. This energy is used to pump hydrogen ions into the lysosome, making it more acidic than the surrounding cytoplasm. The lysosome must work continuously to maintain this pH difference. The lysosome maintains a 1,000-fold higher concentration of hydrogen ions compared to the cytoplasm, creating a steep pH gradient. Chloride channels in the membrane allow negative chloride ions to enter, balancing the positive charge from protons and preventing electrical buildup. The acidic environment is absolutely critical for lysosomal function. Hydrolytic enzymes inside lysosomes, such as proteases, lipases, and nucleases, work best at acidic pH around 4.5 to 5. This graph shows enzyme activity peaks at pH 4.5. Outside this range, enzyme activity drops dramatically. The acidic pH causes conformational changes in enzyme structure, activating them for their digestive functions. The acidic environment also serves as a critical safety mechanism. If the lysosome ruptures and enzymes leak into the cytoplasm, they become inactive at the neutral pH of 7.2, protecting the cell. When a lysosome ruptures, the released enzymes encounter neutral pH and become inactive, preventing cellular damage. Maintaining this pH gradient requires significant cellular energy. The VATPase pumps work continuously, consuming ATP molecules to maintain the proton gradient against leakage. The more lysosomes a cell has, the more energy it needs to maintain all their acidic environments. The acidic pH enables several critical cellular functions. Lysosomes digest damaged or old proteins into amino acids that can be recycled by the cell. During autophagy, lysosomes fuse with autophagosomes to digest damaged organelles and cellular debris. Materials brought into the cell through endocytosis are delivered to lysosomes for processing at acidic pH. Damaged organelles fuse with lysosomes, where the acidic environment breaks them down into reusable molecular components. Let's review how lysosomes maintain acidity and why it matters. VATPase pumps embedded in the lysosomal membrane use ATP to pump hydrogen ions inside, creating the acidic environment. The acidic pH of 4.5 is essential for activating digestive enzymes that break down cellular waste and foreign materials. The pH difference ensures that if enzymes leak out, they become inactive at neutral pH, protecting cellular structures. This acidic environment enables critical functions like protein degradation, autophagy, and receptor recycling that keep cells healthy. The lysosome's acidic environment, maintained by ATP-powered proton pumps, is fundamental to cellular health and recycling.